Hypothesis-driven research is the process of forming and testing and even rejecting hypotheses in order to get closer and closer to the best hypothesis, the best possible answer for a scientific question. It's a very systematic way to do science because the answers are based on data and collecting evidence. But some scientists do what we call descriptive research, which is more exploratory research, when they're trying to collect a lot of information. And the best end result of doing that kind of research is to find a good hypothesis and do hypothesis-driven research. Hypothesis-driven research is an important concept for students to understand because it's the primary way we do science. It's the primary way that we answer scientific questions to understand the natural world and construct explanations that are based on evidence. The first step of hypothesis-driven research is finding a scientific question. And a scientific question is something that you're trying to answer about the natural world. And to be a scientific question, it has to be one that you can answer by making observations, by doing experiments, and by testing it. So, what is your favorite flower is a good question, but it's not a good scientific question because the answers to that question are based on subjective opinions and values. It's hard to test it. So a better scientific question might be something like this. Do butterflies of a particular species prefer to visit flowers of a specific color? That is a better scientific question because it's one that we can test. The second step of hypothesis-driven research is forming a hypothesis. A hypothesis is a possible and tentative answer to a scientific question. And to be a good hypothesis, it has to have three conditions. It has to be specific and clear. It has to be testable. And it has to be, this is really important, falsifiable, meaning the ability to be proven false or rejected. If we think about the scientific question, do butterflies prefer to visit flowers of a particular color? One possible hypothesis for that question is butterflies prefer to visit red-colored flowers. That's something that we can test. I can go into nature or go into the lab and do choice experiments and see if butterflies actually spend more time visiting certain colored flowers. It's also falsifiable. I use my hypothesis to predict what's going to happen in the experiment, and I try to test it, and I try to actually reject that hypothesis. When you set up an experiment and you start with an initial hypothesis, in order to test that hypothesis, we have to rewrite it as two competing claims. And those two competing claims are the null hypothesis and the alternative hypothesis. The null hypothesis predicts in your experiment that there's going to be no relationship between the variables being tested. The alternative hypothesis states that there is a relationship between the variables being tested in your experiment. So for our question about butterflies preferring flowers of a particular color, the null hypothesis might be that flower color has no effect on butterflies visiting flowers. The alternative hypothesis would be flower color does have an effect on the frequency that butterflies visit flowers. So we start with a good hypothesis. That helps us make predictions to set up a well-designed experiment. What is a well-designed experiment? It's one that has a control group, maybe multiple control groups and experimental groups. The difference between those two groups is the variable that we're trying to test. We want those groups to be exactly the same, except the experimental group is going to have that one variable that we're trying to test, red-colored flowers. So if I was setting up an experiment to test my hypothesis, I would want to make sure that all the butterflies were the same species, the same age, they were all equally motivated, equally hungry to visit flowers. I would want to make sure that the flowers I present are exactly the same. I would probably use artificial flowers so I could control smell and what the flowers look like to make everything exactly the same. The only thing I'm going to vary is that independent variable, which is flower color. And I collect lots of data during that experiment. I want to have a very large sample size because what I'm going to want to do is analyze that data using statistics. So if I wanted to test the null hypothesis that there's no relationship between flower color and time spent by butterflies visiting the flowers, I'm going to use statistics to analyze if there is a significant difference between those variables. Are butterflies actually spending more time visiting flowers of a particular color? Or is the variation, the natural variation I might see in the experiment, only due to chance? A statistical analysis will allow me to determine that. Based on the results of that analysis, I'm either going to support or I'm going to reject the null hypothesis. If I reject the null hypothesis, my evidence is supporting the alternative hypothesis. So if the butterflies spend more time, significantly more time, visiting red-colored flowers, then I would reject the null hypothesis that there isn't a relationship between those variables. There actually is a relationship between the dependent and the independent variable. It's important to not be wed to your hypothesis. The process of hypothesis-driven research is to reject hypotheses. It's not about being right. In fact, oftentimes scientists have to go back to the drawing board and come up with new hypotheses because their evidence shows otherwise. So be open to having your hypothesis rejected because you might make a big discovery that you wouldn't have seen in another case.